Bismillah ar-Rahman ar-Rahim. My name is Professor Dr. Mona Gamal Morsi, the lecture about immune response to infectious agent. ILO's integrated learning objectives of innate immunity, the first line of response. At the end of the lecture, I want you to explain the pathways and the fate of hematopoietic stem cell. Then to compare between the first and second line innate and adaptive immunity. Thirdly, to explain mechanisms of innate immunity. Fourth, to distinguish between the role of Toll and node-like receptors in innate immunity. Then to define PAMPs. What are PAMPs? They are pathogen-associated molecular patterns and how do they act. 6, to list cells of innate immunity. 7, to explain antibody-dependent cellular cytotoxicity. Lastly, to list phagocytic cells and how they are stimulated. Now, what are the origin of all immune cells? They are from hematopoietic stem cell, from bone marrow called stem cell. In the exam, this figure will be together. We are going to remove this and put an arrow so that I will ask you, what is this cell? Is it fibroblast? Is it endothelial? Is it epithelial? Or is it stem cell? You are going to choose stem cell. Stem cell is the origin of all immune cell. It differentiates into lymphoid progenitor and myeloid progenitor. The lymphoid progenitor can differentiate in bone marrow into B lymphocyte, which when activated change to plasma cells secreting antibodies. The T cells differentiate in thymus into T helper, T regulatory, T cytotoxic, and so on. And both B and T cell responsible for adaptive or specific immunity. The third, lymphoid cell natural killer play a role in innate immunity. The myeloid progenitor include RBCs, platelets, neutrophil, eosinophil, basophil play a role in innate immunity causing inflammation. Monocytes, circulating monocytes, which differentiate into tissue-fix, macrophage, and dendritic cell. This part is adaptive and this part is innate immunity. What is immunity? Immunity signifies all those properties of the host that confer resistance to an infectious agent. All those properties of the host that confer resistance to an infectious agent. Immunity is two parts. First line of defense called natural or innate or non-specific. The second line is acquired or adaptive, which may be active or passive, and both work to get rid of the pathogen. So the pathogen, when it enters, it will be faced by the first line of defense called natural or innate. If the pathogen is the number or the quantity is too big, it will be faced by this acquired or adaptive immunity. Now, I will ask you to tabulate the difference between innate and acquired immunity. Innate or natural present since birth. It is genetically controlled. The acquired or active or specific or adaptive activated after birth. The innate, the second difference. It does not depend on previous exposure to the pathogen. In the adaptive, it is acquired, depends on previous exposure to a particular pathogen. The third difference, the innate is its action is non-specific, acting on wide range of pathogens. In the adaptive, the action is specific, active against the inducing agent. Lastly, no memory cells in the innate. In the acquired, there are memory cells. What is the function of the memory cell? It can respond to second exposure to the same antigen. So again, the fourth difference between innate and adaptive. Innate present since birth, adaptive after birth. Innate does not depend on previous exposure to the pathogen. The adaptive depends on previous exposure. The innate action is non-specific, acting on wide range of pathogens. The adaptive, its action is specific, acting against the infectious agent, the inducing agent. In the innate, no memory. In the adaptive, there are memory cells, very important to respond to second exposure to the antigen. In the innate is modified by certain factors, age, hormones, and nutrition. The adaptive is formed of two arms, the antibody and cell mediated. Both lines, the innate and adaptive, its function to get rid of the infectious agent. The innate first works, then the adaptive, and then the adaptive activate the innate, and so on. The innate. What are the mechanisms of innate immunity? We have barrier functions, we have microbial sensors, phagocytosis, inflammation, natural killer cells, and the complement system. We are going to discuss each one. The barrier function is the intact skin and mucous membrane, the tight junction in epithelial cells of skin and mucous layer of airway, gastrointestinal, genitourinary act as barrier to invading microorganisms. Microorganisms cannot enter except if there is abrasion or fissure or any cut in the skin or mucous membrane. So first, the tight junction of skin and mucous membrane. Secondly, antimicrobial peptides. For example, present in the epithelial layer like lysozyme, which spreads the sugar from mucopeptide layer of cell wall of gram-positive bacteria, causing its lysis. Defensins, which are located primarily in GIT and lower respiratory tract and create holes in the bacterial cell wall and thus disrupt the bacterial membrane. So the barrier function, the intact skin and mucous membrane, we have tight junction, will not allow the entry of the pathogen except if there is cut or injury. The antimicrobial peptides in the epithelial layer like lysozyme and defensins break down the microbe. Thirdly, the sticky mucus, the mucus in the respiratory, gastrointestinal, the mucus in the respiratory tract act as trapping mechanism, and then the action of cilia, which will sweep the secretion with inhaled microorganisms, either outside through coughing or sneezing, or toward the stomach where they acted upon by HCl. So the sticky mucus in respiratory tract and cilia. Then acidity of the stomach, the HCl will kill the microbe. Then proteolytic enzyme in the intestine, in the small intestine, will inhibit bacteria. The chemical environment, the acidic pH in sweat and sebaceous secretion. The production of fatty acid also on the skin. Bactericidal and fungistatic fatty acid will eliminate the pathogen. So the barrier function include the tight junction of the skin and mucous membrane between cells, the antimicrobial peptide in the epithelial cell as lysozyme and defensin, the sticky mucus and cilia and respiratory tract, HCl in stomach, proteolytic enzyme in the intestine, acidic pH in sweat and sebaceous secretion, and lastly fatty acid in the skin. After microbial, the second innate immunity, first barrier function of the epithelial skin and mucous membrane, secondly microbial sensor. We have two main microbial sensors, Toll and Null. What is Toll? It was discovered by a German scientist in fruit fly. He said, that's a Toll means that's great discovery. So they called it Toll-like receptor. They are group of pattern recognition receptors found on the surface of immune cell and endosomal compartment. So Toll-like receptors are a group of pattern recognition receptors found either on the surface or in the endosomal compartment of many immune cells. They are glycoprotein in nature. They are not lipopolysaccharide, they are glycoprotein in nature. They have an important role in innate immunity. They recognize PAMPs. What is PAMPs? Pathogen-associated molecular pattern. They are highly conserved structure in different organisms. So Toll-like receptor on immune cell bind to PAMPs. The cell surface Toll-like receptor typically sends PAMPs on microbial cell envelope or flagella or lipopolysaccharide of the microbe, whereas the endosomal compartment bind to nucleic acid. So again, what are Toll-like receptors? They are pattern recognition receptor found on the surface of immune cell or endosomal compartment. They are glycoprotein in nature. They play a role in innate immunity, meaning the non-specific immunity. They recognize and bind PAMPs. What is PAMPs? Pathogen-associated molecular pattern. They are highly conserved structure in microbe. The surface part of PAMPs, the microbial cell envelope or flagella or lipopolysaccharide, bind to the surface of toll-like receptor, while the nucleic acid of PAMPs bind to endosomal compartment of toll-like receptor. Binding of PAMPs of bacteria and fungi to toll-like receptor will trigger the activation of signal transduction pathway, resulting in the activation of innate immune cell and synthesis of cytokines as interleukin 2, interleukin 8, and tumor necrosis factor alpha that in turn trigger immune defense as inflammation, fever, and phagocytosis. While binding of viral component to toll-like receptor trigger the synthesis of interferon which block viral replication. This means when PAMPs of pathogen bind to toll-like receptor, it changes the cell from resting to kinetic, producing cytokines as interleukin and tumor necrosis factor, which ends in inflammation, fever, phagocytosis to get rid of pathogen. While if virus bind to toll-like receptor, they trigger the cell to produce interferon, which block viral replication. This is an immune cell, and here toll-like receptor on the surface, and here are bacteria and fungi. When PAMPs of bacteria or fungi binds to toll-like receptor, they change the cell from resting to kinetic. Kinesis means phosphorylation. The cell becomes active, producing cytokines and chemokines which activate other immune cells as TB natural killer. It ends in bacterial lysis. Here the bacteria, here the end lysis of bacteria. How this occurs? By binding of PAMPs of bacteria or fungi to toll-like receptor. When this occurs, it triggers the cell from resting to kinetic. Kinesis means phosphorylation, which end into production of cytokines which activate TB and natural killer. It ends in bacterial lysis. What is NOD? Another microbial sensor called nucleotide binding oligomerization domain, NOD-like receptor. They are specialized intracellular cytosolic receptor. They are essential component of innate immune system and function by the recognition of cytoplasmic PAMPs, resulting in activation of signaling pathways. This results in production of cytokines and chemokines which alert the immune system to the presence of pathogenic bacteria and aid in the clearance of infection. So again, what is NOD? Toll present on the surface and in endosomal. The surface of toll bind to the lipopolysaccharide flagella envelope of bacteria, while the nucleic acid of microbe bind to endosomal toll-like receptor. NOD differs from toll. NOD present only intracellular cytosolic in the cytoplasm and recognized bind only to cytoplasmic PAMPs, resulting also in activation of the cell producing cytokines, chemokines to end in clearance of the pathogen. So toll recognized on the surface and the endosome. Toll on the surface recognize the surface of microbe, extracellular microbe. The nucleic acid of microbe bind to endosomal toll, while not only in the cytosol, in the cytoplasm, recognize cytoplasmic PAMPs, and both of them ends in inflammation, fever, phagocytosis to clear the pathogen. Here the red one toll and the green one NOD. The extracellular organism, PAMPs cell envelope, flagella, lipopolysaccharide bind to the surface toll, activate the cell leading to inflammatory reaction, cytokines and chemokines. If the organism have nucleic acid, bind to endosomal toll. The endosomal here is not present, here only the surface toll. While not only cytoplasmic, bind to cytoplasmic PAMPs, it also leads to inflammatory reaction, cytokines, chemokines, and antimicrobial peptide. Here isolated activation of toll, extracellular pathogen. Here intracellular activate NOD. If the pathogen have extracellular and intracellular pathway, the extracellular bind to NOD the extracellular bind to TOL, while the intracellular, the cytoplasmic, bind to NOD. This potentiates inflammatory reaction, combined activation of TOL and NOD. So in the exam, if you find on the surface, this is TOL. In the cytoplasm, this is NOD. And both of them activate inflammatory reaction by cytokines and chemokines and antimicrobial peptide to get rid of the pathogen. Third mechanism of innate immunity, phagocytosis. What are phagocytic cells? They are polymorph, circulating monocyte, tissue fixed macrophage, present circulating in blood or tissue fixed in lymphoid, liver, spleen, bone marrow, and lung, engulf, its function to engulf microorganisms and remove them from lymph and blood. So again, give me example of phagocytic cell. We have polymorph, circulating monocyte, and tissue fixed macrophage. Its function to engulf and remove the organism from lymph and blood. What enhances phagocytosis? It's called opsonization. What is opsonance? Opsonance, substance which enhance phagocytosis. It renders the antigen more easily taken up by phagocyte. Give me example of opsonance. C3b of complement and immunoglobulin. There are a lot of options. I want two examples. C3b of complement and immunoglobulin because the receptor present on phagocytic cell. So again, what are phagocytic cells? Cells which are capable to engulf microorganisms and remove them from lymph and blood by process called phagocytosis. Give me example of phagocytic cells. Polymorph, circulating monocytes, and tissue fixed macrophage. What enhances phagocytosis? A process called opsonization. Give me example of opsonance, C3b and immunoglobulin because the receptors are present in phagocytic cell. What is this cell? It is phagocytic cell like circulating monocyte, tissue fixed macrophage, and polymorph. What is this process? Is it AGCC? Is it complement? Is it neutralization? Is it phagocytosis? It is phagocytosis. Is it phagocytosis? This picture will maybe come in the exam and ask you what is this process? You should choose phagocytosis. What type of cell can do this? Is it T? Is it B? Is it natural killer? Is it monocyte? It is monocyte or macrophage or polymorph. What is this process? Opsonization. What is the opsin in here? Is C3b part of complement. What is this process? Opsonization. Here the opsonance like C3b or immunoglobulin, it enhances phagocytosis. It renders the antigen more easily taken up by phagocyte. Fourth mechanism of innate immunity, we have inflammation. Injury to tissue initiates inflammatory response which limits the spread of organism. So any injury to tissue by microorganism will elicit inflammatory response to limit the spread of this organism. This response is mediated by many soluble mediators referred to as cytokines. Cytokines may include inflammatory and anti-inflammatory cytokines and chemokines. There will be separate lecture about cytokines, which limit the spread of infection. Many cells are inflammatory cells like polymorphs, like neutrophil, eosinophil, basophil, monocytes are inflammatory cells. And we have inflammatory cytokines and chemokines. There will be a separate lecture for cytokines to limit the spread of organisms. The fifth mechanism of innate immunity called natural killer cells. What are natural killer cells? They are large granular lymphocytes that recognize non-specifically the surface change on tumor cell and virus infected cell and damage these cells without prior sensitization. That's to say, natural killer cells have MHC unrestricted cytotoxicity. That means that natural killer recognize the cell without MHC restriction. It differs from T cytotoxic. We said T cytotoxicity recognize the antigen only when complex with MHC class 1. Here natural killer cell, it binds the foreign viral or tumor or graft cell without MHC restriction called unrestricted MHC cytotoxicity by cell to cell contact or AGCC. antibody-dependent cell-mediated cytotoxicity. They can also bind antibody already attached to antigen on a target cell using their Fc receptor, a process known as ADCC. What is ADCC? Antibody-dependent cellular toxicity. Any cell have Fc receptor could do ADCC, like natural killer cell. Natural killer cell present in spleen and peripheral blood, 5 to 10% of peripheral blood mononuclear cell. So natural killer cell, again, large granular lymphocytes, full of cytotoxic granules. They lack B and T receptor. They have their own receptor. They recognize their antigen without MHC restricted cytotoxicity. They recognize it by cell-to-cell contact or ADCC through its Fc receptor. It lacks memory cell, no memory cell. Here, virus infected cell. The virus produces its epitopes or antigen. Here the antibody by Fc receptor, it binds to natural killer. Then the natural killer releases its perforins and granzymes, activating caspases. Programmed caspases produce apoptosis, programmed cell death. This animation here, virus infected cell. The virus produces its epitopes, the most antigenic determinant. Here circulating immunoglobulin bind to Fc portion of natural killer cell, which release its perforins and granzyme, the cytotoxic granules, which activate caspases, leading to apoptosis. Here, destruction of virus infected cell by natural killer cell through ADCC. Which cell can do ADCC? Any cell which have Fc receptor, like natural killer cell. There are here natural killer T cells, not present in your book. Here it is, they are rare types of T cells that possess, they are different from the natural killer. They have receptors for both natural killer and T cell. So what are natural killer cells? They are rare types of T cells that possess both receptor of T cells and natural killer cell. They are the only T cell which work in innate immunity. Their cytotoxicity is unrestricted by MHC, and they lack memory cells. So this rare types, we should know that all T cells work in adaptive or specific, except this rare types of natural killer cell. Natural killer T cell, they have both receptors of natural killer and T cell. They are the only T cell which work in innate immunity. Its cytotoxicity not restricted by MHC, and they lack memory cell. The last mechanism of innate immunity are the complement system, another key of component of innate immunity. Which pathways of complement work in innate immunity? Which pathway? We have three pathways. Here only two works in innate, the alternative and the lactic. The alternative come in contact with the microbial pathogen. It starts by C3. The lactin come in contact with microbial mannan, start by C4. And the alternative factor B, D, and propergine works, while the lactin C4 and C2 works instead. Both these pathways play a role in first line and defense before antibody formation and provide immediate protection against microorganisms. So again, to summarize, at the end, I wanted to explain the pathway and fate of hematopoietic stem cell. It is the origin of all immune cells, which differentiate into lymphoid and myeloid. To compare between innate and adaptive. To explain mechanism, we have 6 mechanisms of innate immunity. We have the barrier function, skin and mucous membrane. We have microbial sensor, mainly Tal and Nod. We have phagocytosis, inflammation, complement, and we have natural killer cell and natural killer T cell. To distinguish between Tal and Nod, Tal on the surface and endosomal, not only cytoplasmic. Tal recognize the surface on the pathogen, and the nucleic acid of the pathogen is bind to endosomal, not cytoplasmic PAMPs only. Define PAMPs, highly conserved structure in bacteria, virus, and fungi, and it bind to Tal and Nod. to produce inflammatory fever phagocytosis, and it ends in the elimination of microbial pathogen by cytokines, chemokines, and antimicrobial peptide. List cells of the innate immunity. What are cells playing a role in innate? We mentioned phagocytic cells, natural killer cells, inflammatory cells, and natural killer T cells. Phagocytic cells, natural killer cells, natural killer T cells, and inflammatory cells. Explain ADCC. Any cell have Fc receptor could do ADCC like natural killer cell. It's a cytotoxicity not restricted by MHC. List phagocytic cells like polymorph, circulating monocyte, tissue fix, macrophage. How do they act? They are enhanced by opsonins. Give me example of opsonin. C3b and immunoglobulin. Thank you. Distal on the surface. This not intracytosolic. This is phagocytic process by monocyte, macrophage, or polymorph. This is opsonization, and this is ADCC by natural killer cell. Thank you.